Okay, so now we start our unit on cells. So we're going to start with the very basics. We're going to be talking about cell structure and function. So to start with, it's important that you know that the cell is the smallest unit that is capable of performing all the functions of life. And remember, we mentioned those in the last lecture. So bear those in mind as we move forward and start talking about our cells. So if you look at this scale on the right, it kind of gives you a perspective of about how big cells are. So starting from the very left side, they're the atoms, amino acids, proteins that we talked about. When we get to the 10 nanometer range, those start to become viruses that we mentioned are not living things. Sliding to the right, we get into the organelle level, and we're going to talk in detail about organelles within a cell. The particular one they show here is a chloroplast, and that's about 1 micron. And then we get into cells. So we have bacteria cells and plant and animal cells. And so that's in the range of anywhere from 1 to 100 microns. And if you look below that, you'll notice that that's about the range of a light microscope. So prior to microscopy, most cells could not be seen with the human eye. So our typical range for cells is somewhere between 5 and 50 microns in diameter. There are exceptions that are a little bit larger, like egg cells. And one thing I'm going to point out is that this scale across the top is what we call a metric scale. And so it is going to jump in units in base of 10. And you're going to be learning more about that in lab. Our first lab is centered around the metric system and measurements with it, and just kind of some basic lab techniques. So there is what we call cell theory, and it is composed of these 3 points. One is that all organisms are composed of cells. So without a cell, you don't have a living organism. And like I mentioned in the previous slide, cells are the basic unit of life, so it can perform all the tasks of a living organism. And then the third one is that cells come from pre-existing cells. They do not come from nowhere. And we're going to take a little bit of a look at the evidence, particularly for that portion of cell theory. So for some of the first evidences for cell theory, we relied on microscopy. So microscopes greatly increased man's ability to visualize tiny objects. So this scale at the top you've seen before in the previous slide, but I just wanted to point out that even when we have the light microscope that allows us to see whole cells, when we get to electron microscopes, that even increases our ability to see the details of within the cell. And I will argue also what's not on this is that we also have atomic force microscopes that allow us to look at the atomic level. But using our microscopes, every living thing, when we put it under the microscope, is composed of cells, and you can see that visually. Now, there are some cells that don't conform to what we typically think of as a cell. For instance, our muscle tissue, our muscle cells, some of them have multiple nuclei, or tracheids within a plant. There are certain types of tracheids that don't have any nuclei whatsoever. They actually are always paired with what's called a companion cell, who regulates their protein synthesis and all of that. But every living thing is made up of cells. So the experimental evidence that we have for cell theory really focuses on that third point, and that is that we don't spontaneously generate new cells. Cells have to come from pre-existing cells. So our 3 lines of evidence for that based on experiments is that cells that were removed from tissues can survive independently for a short period of time. Secondly, nothing smaller than a cell is able to live on its own. And then thirdly comes down to experiments performed by the Italian scientist Redi and by Louis Pasteur. And that was evidence that supported that that cells do not spontaneously generate. So Redi's experiment was way back in the 1600s, and at the time it was thought that maggots spontaneously grew from meat. So you would be getting insect cells straight out of meat. And Reddy disproved this by taking meat, sealing it in a sterile container, and showing that no flies appeared. Now, when he left it uncovered, of course, the flies would appear, and then also when he covered it with paper, the flies were attracted to it and would lay their eggs, but those eggs could not grow because they actually feed on certain proteins in the meat once they hatch. Now what Pasteur did was slightly different, but proved the same point, and that is that he took broth and boiled it, and as we all know, boiling it kills all the bacteria, and he had a curved neck, basically meaning that things could not come into it. Once that broth was boiled, the steam went off and there was nothing getting sucked back in. And when that happened, there were no microorganisms in the broth. It remained free of them. Now, when we took that neck back off, then all those bacteria in the air could come into the broth and begin growing microorganisms again. So that's our experimental evidence for cell theory. So just for review, a cell is the smallest building block of life. It is the smallest form of life that can, so it can perform all the functions of life on its own. And then it does not spontaneously generate. Cells have to come from pre-existing cells. So when we're talking about cells, or actually all of life, we want to categorize them. Normally that's called taxonomy. We start to categorize them in order to help us understand the differences as well as the relationships between different organisms. So at sort of the largest umbrellas that we can divide life into is what we call the domains. So there are 3 domains that life is divided into as far as science is concerned. And these are based on the types of cells that are in those particular domains. So the 2 types of cells that we're going to talk about today are in general, but are prokaryotes and eukaryotes. So our domains are based on that loosely. So if you'll look, we have our eukaryotic cells, and we're going to get into more detail about that in the next lecture. And under that domain eukarya, we have our protists, our fungi, our plants, and our animals. The traditional things that you know about. And then under this right here, this is where we have prokaryotes. And those prokaryotes are divided into 2 domains, bacteria and archaea. All of these, when we start talking about the different parts of cells, one thing that comes down to very basics is that every cell has ribosomes with ribosomal RNA. And because everybody does, that ribosomal RNA is used to categorize organisms. Okay, so why 3 domains? And some of this is going to make a little more sense to you as we go into exactly what these different parts are of the cell. But in this Venn diagram right here, the first thing we're going to look at is what do all of these 3 domains have in common? So that would normally be in this section right here. So what all living things have in common is they're made up of cells. They have DNA or RNA or both, some sort of genetic material. They have a cell membrane or a cell wall. They have ribosomes, that's what I talked about a minute ago, and they have a cytosol or cytoplasm. So when we start looking at the differences between them, let's start with our eukarya right here. So eukarya have a phospholipid bilayer that makes up their cell membrane. They have a nucleus and they have what's called organelles. And we'll go into detail about what organelles are, but things that you're familiar with, mitochondria, chloroplasts, that sort of thing. Then we're going to come down here to bacteria and archaea. And bacteria and archaea are composed of prokaryotic cells. Remember, the eukarya domain is made up of eukaryotic cells. Well, bacteria and archaea are made up of prokaryotic cells. Now, there's a slight difference between them in that bacteria contains what we call peptidoglycans in their cell walls, and we'll talk about that a little bit. And then the archaea prokaryotes have a monolipid membrane, and they are the ones that live in harsh environments. So if you think deep sea thermal vents, that's where you're going to find your archaea prokaryotes. Okay, so let's jump into our prokaryotic cells, also what we call the prokaryotes. So as I mentioned, they're contained in domains bacteria and archaea. And prokaryotes themselves, that word actually means before the nucleus. So if people are thinking on the macroevolution scale, they think that prokaryotes came first and they came before the cells that have a nucleus. They are single-celled organisms, and they lack any of the membrane-bound organelles. Now, how they acquire energy varies tremendously. They can be photosynthetic, they can be autotrophic, they can be chemotrophic, heterotrophic, and so there's a large variety of prokaryotes. So let's take just a moment to look at the structure of the typical prokaryotic cell. It kind of falls into three different categories for what the cell is made up of. The first is the cell envelope, and that envelope contains the plasma membrane, which regulates what comes in and out of the cell. And since there are no organelles, there are invaginations in the plasma membrane where processes such as respiration or photosynthesis occur. And then there's a cell wall, and that provides rigidity, some stability, and is especially important with fluctuation of water pressure within the cell, so it provides that stability. And then some bacteria, or some prokaryotic cells, but not all, have an additional layer called a glycocalyx. Now, that glycocalyx can be very well organized, such as in a capsule, or it can be very loose, and then that's what we think of as a slime layer. So that's the cell envelope. The cytoplasm, of course, contains ribosomes, because we know that every living thing contains ribosomes. Thylakoids, which are specific to cyanobacteria, and then a nucleoid. And that nucleoid is basically a concentrated area of DNA. Remember, it doesn't have a proper nucleus. And then the third category is the appendages. It can have a flagella, a conjugation pilus, or a fimbriae. So this is a closer look at the cytoplasm. So like I said, it contains ribosomes, that's for protein production, and then contains a single nucleoid, where it has concentrated DNA. So if you look at our cell right here, that would be this right here. Interestingly enough, that DNA is a single chromosome tightly wound together. So you can see here in our cell on the right, if you extend it out, that's that single chromosome, all tightly wound together to produce that nucleoid. They also have plastids, which are extra rings of DNA, and we'll talk a little bit more about that, but here they are right here. And then the other thing is that if it's a cyanobacteria, it has thylakoids. And we're going to talk about that a little bit later because we want to discuss how cyanobacteria is thought to have played a huge role in Earth's history. So for now, just know that thylakoids are stacks of discs used for photosynthesis, and those are present in autotrophic prokaryotes, particularly in cyanobacteria. So here is a picture of those plasmids that we talked about. Those are just accessory DNAs, and they're usually circular and very small, and they just contain extra genes. And sometimes this is where the genes are located for antibiotic resistance, so they do play an important role. So as we discussed, there are three types of appendages associated with prokaryotic cells. There is the flagella, which is towards the one end, and as it circles around in 360 directions, it propels the bacteria forward, much like a propeller would work. The fimbriae are these small hairs on sort of throughout the sides, and they usually help with attachment to a host if it is a parasitic bacteria. And then there's the conjugation pilus. That is actually not in this picture. We will go on to the next slide to show you what that is. So here we go. On this one, here is our conjugation pili, pili being more than one. But if you notice, the conjugation pili comes from the cytoplasm and out. And what that is used for is to allow for transfer of DNA from one bacteria to another. So that's what that's for. And again, you can see the fimbria here and our flagellum here. So those are three types of appendages that we see on a prokaryotic cell. So since we mentioned these appendages, let's talk about how prokaryotic cells move. Most of them do move using the flagella appendage. So a flagella is composed of a protein called flagellin. It's wound tightly in a helix and anchored into the cell by a basal body. Sometimes that's also called a kinetosome. And that basal body is made up of microtubule structures near the cell surface. And it, like I said, it allows that flagella to rotate in 360 degrees and allows the bacteria to move forward just like a boat propeller. It's important to note what I have here in blue. Prokaryotic cells do not have cilia, so don't confuse the fimbriae that we looked at before with cilia. Only eukaryotic cells have cilia, and they use those for motility, but prokaryotes only have flagella, not cilia. So here's just a close-up look at the flagella of a particular bacteria, just so you can see where it all attaches. So here's the glycocalyx on the outside, the cell wall, and then the plasma membrane. And so the anchor really, the basal body, does really go all the way through into the membrane and allows for that flagella to rotate. Talked in generalities about flagella. There's a difference with our flagella within the domain bacteria and the domain archaea. Remember, they're both prokaryotic cells. The flagella in bacteria is what you have pictured on this slide. It's thick and the basal body is present. So the basal body, if you look at these micrographs over here on the right, this is a cross section through one. Each of these little sections here are made up of a triplet of microtubules, and there are 9 of those. And that's very specific to bacteria basal bodies. That's how the flagella is attached. The prokaryotes in archaea, the flagella is very thin, and they actually lack a basal body. Instead of having a basal body, there is a flagellin hook that attaches the flagella. So that's the difference between those two domains. So how do prokaryotes reproduce? Their reproduction is asexual, and it's through binary fission. So prokaryotes are haploid, meaning they only contain one copy of the chromosome. So that is specific to prokaryotic cells. So let's talk a little bit about this binary fission. Okay, so since we only reproduce asexually, how does recombination occur? Remember, recombination is what allows for the introduction of new genes and the possibility to adapt to your environment. Well, this happens in 3 different ways. The first being conjugation, and that's where the pilus appendage that we talked about comes into play. So remember, it comes straight out from the cytoplasm and through the envelope. So it is literally, DNA is donated from the donor cell all the way to the recipient cell. And so a sharing of DNA occurs that way. That's conjugation. The second way is transformation. Transformation is when the cell picks up free pieces of DNA either from living cells that have secreted it or from dead cells as they break down and their DNA is released into the environment. Then these bacteria can pick it up. The third way is transduction, and that's when the bacteriophage carries portions of the DNA from one bacteria to another. So remember, the bacteriophage inserts viral DNA into bacteria, into the bacterial DNA, and when it's clipped out, a piece of the bacterial DNA may be clipped with it, and then it's transferred into new bacteria, so that allows for the new possibilities of genes. So now that you know some characteristics of the prokaryotic cells, let's just look at the differences between a prokaryotic cell versus a eukaryotic cell. And this particular picture, these are not to scale. The prokaryotic cell is much smaller than the eukaryotic cell, but it's enlarged just so we can take a look at the differences between the two. So if you look to the left, of course, we have the appendages that we've talked about and that nucleoid in the middle, that concentration of DNA. And then if you look on the right at the eukaryotic cells, you'll see lots of membrane-bound organelles. So it's very organized and much more complex than the prokaryotic cell. So as we said, prokaryotic cells are in the domain bacteria and the domain archaea. So we're going to look at both of those today. And let's start out by looking at the domain bacteria. So there are a couple of things here. Their cell wall is composed of a peptidoglycan. They come in 3 different shapes, and they're categorized based on their growth arrangements. So here is just an image of the peptidoglycan. Most bacteria have cell walls composed of this, and they are linked, it's basically polysaccharides linked by amino acids. It's very rigid and provides structure for the bacteria. Bacteria come in 3 cell shapes, and they're very distinct, and it's often how we identify what type of bacteria it is. So the one on the left has a spiral shape to it. This you can only see in 2 dimension really, but 3-dimensionally it has a spiral effect, and so that shape is called spirillum. The second one in the middle is more of a capsule or a rod shape, and we call that bacilli. And then over to the right they're actually like spheres linked together. Well, in this case they're linked together, but they're round spheres, and they're called cocci. Okay, so then now we have 2 ways of starting to classify them, and the third way is their growth pattern. So the way we say that is that if you look at this first one, and Staphylococcus, staph means that it's arranged in clusters, and remember coccus or cocci were round shaped. So based on bacteria names, you usually know a little bit about the bacteria. So Staphylococcus is cocci-shaped bacteria grown in clusters, and that's what that name means. The second here, the Streptobacillus, strept means that it's a chain of bacteria, and bacillus is just what it sounds like. That would be the rod shape. So you have a chain of rod-shaped bacteria. And then down here at the bottom we have Diplococcus. Diplo, just like you would think, means that it's pairs, two of them at the same time. And again, there's that coccus, which means they're round, so round pairs. And that, by the way, is usually what causes pneumonia, in case you were wondering. So that is how we tend to name the bacteria that we are looking at. I want to draw your attention to a particular type of bacteria called cyanobacteria. And it is still in the domain bacteria. But it's significant on Earth because this particular group of bacteria typically lives in the top layer of bodies of water, and they provide two main functions. They are primary producers, meaning they're eaten by many animals, and so they start the food chain. The second thing is that they produce a lot of oxygen, quite a bit more than you might even think. And so the way that they do this is they photosynthesize like plants do. And in addition to chlorophyll A and B, they oftentimes have accessory pigments that will give them different colors. So some may even be black or yellow. And they photosynthesize in the same manner as plants, and they contain thylakoids. So this bacteria has an inner membrane that has thylakoids. And they can grow in these three different growth patterns. They can either be single-celled, they can live in colonies, or they can be what we call filamentous. And if you come do Bio 112 with me, we'll take a look at those in depth in the lab. So this is one cyanobacteria called Glioiocapsa, and as you can tell, it lives in a colonial form. And so Glioiocapsa actually lives just in moist environments. This one's actually not in a body of water, so this is like in really moist soil, like if you have puddles, something like that. And oftentimes this cyanobacteria lives in a mutualistic relationship with fungi in order to produce lichen. So if you know what lichen is and if you've seen it in the woods, this is the bacteria portion of the lichen. Here are a couple of filamentous cyanobacteria, so I just wanted you to see a couple of examples of that. Oscillatoria is basically, it looks like a straw, a very flexible straw. And then for Anabina over here, we have two cell types in their filament. So most of them are going to be vegetative, but they have what we call heterocysts right here. So there are these circular cells that you see every now and then. It almost looks like a necklace with a little pearl in the middle. And those heterocysts are important because they function to help the bacteria with nitrogen. So those cells are nitrogen-fixing cells. And so Anabina is able to photosynthesize and to fix nitrogen, which basically what that means is changing it to a form that is usable by, in this case, the cyanobacteria itself. So fascinating examples of filamentous cyanobacteria. Whew, okay, we've made it through our domain bacteria. Now we have the domain archaea, which is our second domain of prokaryotes. And these usually fall into the categories of being anaerobic. They can be thermophilic, which if you remember what your Latin roots, thermo being heat, philic, they love heat. And most of them are chemoautotrophic. And there are three different types of our archaea. A lot of times they're referred to as archaea bacteria. So if I say that, that's why. But there are three main types. There are methanogens, halophiles, and thermoacidophiles. So let's talk about those. So I want to start off by looking at the plasma membrane of our different types of cells real quickly. Our domain bacteria and our domain eukarya have what we call the lipid bilayer plasma membrane. So if you'll remember when we were talking about phospholipids in our organic chemistry unit, we have our hydrophobic tails and our hydrophilic heads that come together to form that bilayer, and there's always seems to be this slight space in between the two. So that is in our domain bacteria and our domain eukarya. Well, in some archaea, and I would argue most actually, there's no bilayer. There's a lipid monolayer. So we still have the hydrophobic tails and the hydrophilic heads, but they link the tails together, and that allows for some rigidity and some protection. So the archaea, or archaebacteria, that's the other thing that it's called sometimes, they live in extreme environments, so high heat, high salt, so they need a membrane that can certainly protect them from their external environment. So that's the reasoning that we think that they have this lipid monolayer. So let's talk about our methanogens to start with. They are obligate anaerobes, so that means that they require an environment that lacks oxygen, and they produce methane gas. So they live in swamps, landfills, and they live in the intestines of humans and cows. The second type is a halophile. Halophiles are aerobic, so they do require oxygen, and they are chemoheterotrophs. So they tend to live in very salty places, because halo meaning salt, phile means they love salt. So they live in very, very salty places. So like the Dead Sea, 7 times saltier than regular seawater. And this is where the halophiles live. The third type are the thermoacidophiles. So just as the name suggests, they like it very hot and they like acidic conditions. So the geysers and hot springs fit that bill, and there are lots of them in Yellowstone National Park. If you've had the opportunity to go out there, you know it's beautiful, but it's also stinky because it has a lot of sulfur production. So this image right here is of the Grand Prismatic hot spring in Yellowstone, and this is looking from the top down. It's an aerial view. So just to give you an idea, this right here is a walkway, and right here are 2 people. So that just gives you the scale. Now, interestingly, in these springs and in the geysers, depending on the depth of the geyser, the deeper you go, the hotter it is, and sometimes the more acidic. So depending on what environment the archaea bacteria desire, you can tell which ones are present, because they're what causes the color of the water. So those that can handle it really, really hot create this dark blue. As you get closer to the edges, it gets a little more greenish yellow, and that's slightly cooler, still too hot for us. But that's where the different thermoacidophiles live, and they make this beautiful picture. So here is a geyser from our family trip to Yellowstone one time. This is one of my daughters, my youngest actually. Happy as a clam to be here. You can see how beautiful the geyser is due to those lovely chemoautotrophs back there, the archaea. And so what I probably failed to tell you, sorry about that, is that these chemoautotrophs, these in particular, use hydrogen as an electron donor, and they use sulfur as an electron receptor. So the byproduct is hydrogen sulfide, so that's what makes it so stinky. But my little one here could care less about the stink. She thought it was pretty and lots of fun. But now I have to show you the next. This was one of my other daughters. And you like the smile? It's a fake smile. Because she hated the smell, absolutely hated the smell of these geysers because of the sulfur production. And so she would go, "Do we have to go there?" And I'd say, "You have to give me a smile for the picture." So anyway, so that's my end for this one, just to give you a little laugh. But those are the thermoacidophiles, making one daughter happy and the other not so much. Anyway, Thanks for listening today, and we will get back together soon.